My name is Máté Gyimesi, and we are working at the Department of Biochemistry, and we focus on preclinical drug development targeting motor proteins. 4 years ago, when I won the Promising Researcher of ELTE, I finished my interview with a motivation statement that I'm devoted to our project because we believe that we have a molecule in hand which at one day will be administered to a human patient with severe muscle spasticity. Since then, a lot of great successes have been achieved at different levels. First, following the international trends, we established a startup company together with András Márnási Csizmadia at the Department of Biochemistry here at ELTE. This company is a preclinical drug development company focusing on motor proteins. We would like to target these motor proteins to cure severe diseases. With Motor Pharma, we achieved our first success when we could buy and license the development and marketing rights of the MPH inhibitor family, which contained the lead molecule MPH220, which is now a drug candidate. So MPH220 is a first-in-class muscle relaxant which directly targets the effector protein of muscle contraction. It's important because current muscle relaxants act on the nervous system and target upstream regulators of muscle contraction. Therefore, they have a lot of side effects, including cardiovascular and neurological side effects. Most importantly, these molecules are with very low efficacy. More than 60% of the patients treated with these molecules simply don't respond to the treatments. In contrast, MPH220 directly targets the effector protein of muscle contraction. Therefore, the efficacy is 100% without the cardiovascular and neurological side effects. Scientifically, the great challenge in this development was to find a selective inhibitor which is selective for the skeletal muscle myosin. It's important because human body, human cells have 5 different isoforms of myosin 2, which are structurally very similar to each other. In the binding pocket for the inhibitor is practically identical. And we could identify and target a single residue change in the binding pocket, which resulted in MPH220, which is absolutely selective for the skeletal muscle myosin and doesn't affect the cardiac myosin or the smooth muscle myosin. The importance of this project was acknowledged by its publication in Cell, which is the most prestigious biological journal, and we could publish its preclinical efficacy studies, including the in vivo animal models. In these animal models, we generated a nervous system injury in the rat's brain and developed spasticity, which was very similar to the human situation, where the whole body is stiff and the movement of the patients or the movements of the animals in the animal models was very severely affected. The legs couldn't really move because of the stiff muscles. However, after treatment of these animals with MPH220, relaxed the muscles and the gait functions came back to the normal, to the healthy level. Following the publication in Cell, we experienced a vibrant investor interest. These negotiations ended in a formation of a US spin-off company called Motric Bio, which was established by Motor Pharma. Aditum Bio is a biotech investment company established by Joe Jimenez and Mark Fishman, who were the CEO and the research president of Novartis for more than 10 years. These pharma giants selected our project, MPH220, from the more than 2,000 investigated projects, which can make us really proud, I guess. So in Motric Bio, now we focus and work hard on the clinical development, the human clinical development of MPH220, and the first in human clinical trials will start early next year. And we can be very optimistic about the future career of this molecule because of the fantastic toxicological and pharmacological properties, because as I said, it's very potent, it's very selective, therefore it's very safe. And due to the highly professional clinical development team behind the project in Motric Bio. Our major focus now is to improve stroke regeneration by relaxing the vasospasm generated inside the brain after an ischemic stroke effect. And we also investigate possibilities in the tumor growth and metastasis field if it's targetable by the ectomyosin system. So we hope and we believe that in the next few years we will be able to generate new successes as we did before with MPH220.